Today's topic is the central dogma. Central dogma is basically just transcription and translation, which means your DNA is creating mRNA and the mRNA then travels to the ribosome where it is translated into a protein. And that is the central dogma. You should have already read the basic vocabulary. I am not going to go into many details about rRNA, ribosomes, tRNA, codon, anticodon. I'm going to assume you know all those vocab terms. I'm not going to teach those to you right now. We're going to use them. If you don't know those vocab terms, please return to your textbook and read and get the vocab down and then come back and watch the video. Okay, we're going to start off just with a few basics that are important for us in AP Bio that you may not have encountered before. So I'm going to try to keep this just to what we need to know. So whenever you are having a transcription event occur, there's three phases. There's initiation, and then there's elongation, and then there's termination. So initiation, elongation, termination. Okay. These three phases occur in both transcription and translation. Of course, they're different between the two, but the terms are the same. Okay, so initiation, that's an I, elongation, and termination. Okay, in transcription, this is where we're making mRNA. This is occurring in the nucleus of eukaryotic cells. Remember, prokaryotic cells don't have a nucleus, and so clearly it's not going to happen in a nucleus. Instead, it happens just with the genetic material, which is in the middle of the cell, nucleoid region. Okay, we're looking for a promoter in the initiation, and so in most cases, especially in eukaryotes, we're going to be looking for what we call a TATA box. Okay, and that's just simply thymine, adenine, thymine, adenine, repeated. Okay, and so this is where RNA polymerase, that big enzyme that does all the work, RNA being the molecule, polymerase, polymer being what it's making, like a long strand of monomers, and then ACE is referencing to the fact that it's an enzyme. So ACE, A-S-E at the end of a word refers to the fact it's an enzyme, and an enzyme is a protein that helps to lower the activation energy, lower the energy required to do a particular task. And so this is RNA polymerase. So it's an enzyme that's making a polymer of RNA. Okay. An RNA polymerase comes in and it can find this. It's basically chemically scanning for a promoter region. And so for that is the TATA box. Okay. And it's going to be, depending on the gene it's looking for, it could be on either side of the DNA strand. So when the DNA is opened up, like a ribbon being opened up, then it could be on the top or the bottom. It depends on what gene it's looking for. We have multiple terms for these strands. And so to me, it's just nice to think of the template strand is the one that is coding the mRNA out. Like the DNA template is being read, and like with base pairing rules, it'll create an mRNA on that template. So then the other strand that's open but not being acted upon to create an mRNA at the time, could be another time, but not this time. That strand I would just call the non-template strand. However, the template strand has three names besides template strand. And these are ridiculous. Honestly, it's kind of why I'm sort of giggling. But basically you have the non-coding. That's the one that throws kids off the most, which means the one not the template strand, the other strand, is actually called the coding strand, even though it's not coding mRNA. Do you know why it's called that? Because the sequence of that strand is almost identical to the mRNA strand. So it's almost the same code. The only difference being there will be uracil or U's where there would have been T's in the DNA. Okay, so that then we call that the coding strand, the one that's actually not coding. Anyway, so non-coding is, so these are all synonyms for template. Okay, so these are synonyms for template. So we have the non-coding strand is synonym for template. We also have another weird one, the minus strand. I don't even know where they're getting this stuff, to be honest. And then the anti-sense, which means the other strand is the sense strand. You know what? Sometimes biologists need to just get a hobby. So these are all the template strand. Now, oops, that went right off the edge. Now, that being said, who cares? It says template. Okay, just deal with it. I don't want to mess with it. That being said, the only ones that I have seen used on the AP Bio test are the template strand and the coding strand, which is the non-template strand. Whatever. Okay, so those are the template strands. Okay, cool. Spent plenty of time on that. So in the elongation phase, all that's really happening is the RNA polymerase is putting matching base pairs. Like if there's an A in the template strand, then it'll put a U on the mRNA, and the mRNA is just growing. Okay, the mRNA is just growing longer and longer. That's what they call it, elongation. It's just growing longer and longer. And then the termination phase, it comes to a series of bases that are the series of nucleotides, let's use the real words, that are signaling for it to be done, to terminate. Okay, and so then it'll stop. The RNA polymerase will leave, will disassociate. Of course, the DNA is wound back up, so all is well. Okay, so this is the end of it, and this is just building the mRNA. These terms are all in your book. Please don't just rely on that tiny little description. You're going to need to know them more. Okay, now cool. So we have an mRNA created, and then there's two things you need to know is that eucs and procs do things different at this point. Up until this point, it's been pretty much the same. RNA polymerase is different. It's like a different one. It's like RNA polymerase 1 and RNA polymerase 3 that are used for building the mRNA, but otherwise, everything's the same, okay? Until now, okay? Eukaryotes, remember, these are the ones that have a nucleus and all that stuff. We have RNA processing that has to take place. And it has to be completed before translation begins. Okay? So, cool. Prokaryotes, no processing. And then translation can begin before transcription is even done. Weird. I don't know if you can see that or not. Sorry. Okay, so they can actually be translating an mRNA strand in prokaryotes when they haven't even finished creating the mRNA strand completely. We don't do that in eukaryotes. Remember, these are the ones with the nucleus. Sorry, these are the ones with the nucleus right here. We don't do that. It has to be completely processed and finished before it can ever be translated. Prokaryotes, a little different, a little faster, okay? So mRNA processing, we're going to go, so this is eukaryotes only. So mRNA processing, I'm really being messy today. I don't know what's wrong with me, sorry. So for eukaryotes only, we're going to see them. First of all, you have your little mRNA here, okay? There are places in it that are not needed to create that protein. There are places in the mRNA that are used for gene regulation to decide what's going to be transcribed and what's not. There's some places they can't even really figure out what the heck they're for, but they don't need those areas of the mRNA. So if this is the mRNA and it's just full of like nucleotides, there will be areas in it that are called exons. I'm just going to highlight it like I'm highlighting a strand. So those are A's and U's and G's and C's. So those are actually like codes right there. Like I'm not just highlighting some weird strand, but it's codes. And then there's other A's and U's and G's and C's. Okay. But they're not actually like parts that are going to become a protein. And so what does it do? It splices. Okay. It splices out segments called introns. Okay, so introns are out. Ins are out, right? So let's pretend like the blue is the intron. Okay, so the blue is the intron. Okay, so it's going to cut out that, and it's going to cut out that, and it's going to cut out that, and it's going to cut out that. And then all you have left after all that's cut is just the red. Okay, and those segments are called exons. So all that's left is exons, and that's because that X, the EX, is like expressed. Okay, that's what the exon comes from. So that means that's the part of the mRNA that's expressed into protein, and that's why it's called exons. Also, on eukaryotes only, after that's done, on the 3' end, it makes a poly A tail, literally a bunch of A's, poly A, many A's. And then on the other end, it puts on what we call a 5' cap, otherwise known as a GTP cap. Okay, guanosine triphosphate cap. Okay, so that's what goes on this end. So on the 5' end, and I don't know which is 5 and 3, it doesn't matter. But on the 5' end, you get a 5' cap, and on the other side, you get a poly A tail. We think that's for protection of the mRNA as it moves through the cell so it doesn't get degraded, but we really don't know because eukaryotes can do just fine without those parts. So we're not really exactly sure why we have those particular parts. That's called RNA processing. Also, funny story, something called alternative splicing can happen. Completely different. This does not happen all the time. So alternative splicing happens when maybe it only splices this one out right here. So maybe it only splices out that one, and then maybe it just splices out a chunk of that one. So same gene, same mRNA strand, spliced differently is translated differently. So now through alternative splicing, you can have two different proteins. And they're probably going to be closely related proteins, but they'll be different. Weird. Okay, cool. Moving right along. Let's get out of this nucleus. Okay, so here we go. Now we're going down to the ribosome, which you should already know is made out of rRNA. Look that up if you don't know. Here comes our cute little mRNA hauling through here. Remember, it's made up of all kinds of like different. There we go. I just can't talk and do this at the same time. It's made out of all different nucleotides. tRNA over here basically comes in. Whoa, what the heck shape is that? I don't even know. Here's our tRNAs. Okay, this is called a codon. Yeah, 3-base sequence, 3 nucleotide sequence, codes for an amino acid. An amino acid is a building block of a protein. Okay, an amino acid is the monomer of a protein. Look at the codon table. Look at the codon table. Isn't it nice? Okay, you will see that there are many codons that code for about 22 different amino, stretching my brain, 22 different amino acids, or is it 23? But there's like 64 different codon sequences. And so clearly there are some codons that code for the same amino acid, like 2 or 3 codons. But I squared in AUG. Look at your thing. Look at your codon chart. Find AUG. What does it code for? You should have found methionine or MET. MET is the universal start codon. I kind of made that noise because there are a few that, of course, exceptions that prove the rule, but we're going to say that is it. AUG is the start codon. In the initiation phase of translation, this is translation, you're translating your mRNA into a protein. In the initiation phase, then you will begin whenever the ribosome or RNA in the ribosome connects with the mRNA as an AUG. It's time to start, okay? So then methionine will be, there he is. There's methionine right there. How did we know he's coming? So this is the amino acid right here. So that's the amino acid. Oh my gosh. My little projector is just too tight of a screen. So that's the amino acid that's coming in. This is the codon. The anticodon. Yeah, see? U and the A pairs with U. C pairs with G. Anticodon. Codon, anticodon. Codon chart, no anticodon chart. Okay. So then it reads that and it's like, yay, I need to bring my amino acid. And so it brings it in. Okay. That is elongated, meaning all the polypeptides will start to be attached to this. Watch the video, watch the animation. It's pretty cool. So then the polypeptide chain grows because it's like matching up the correct codon with the anticodon until it's finished. And then when it reaches a stop sequence, which you can see on the codon chart, it reaches a stop sequence, then it will be terminated. And the polypeptide will then go into the next process we'll talk about later, which is protein folding. And it will be, then it will be like all finished, and it will be processed, and the protein will be ready for use. Okay. I think that should about do it. Hope that helps. Have a great day, and I'll see you soon.